The figure 14-9 is showing you just an illustration of all the different innate defenses, physical barriers, phagocytes, immune surveillance by natural killer cells, interferons, complement, inflammation, and then fever. So this is a really good summary of the body's innate defenses. It could actually take place of just about everything we covered with the innate defenses. So checkpoint questions. What types of cells would be affected by a decrease in the number of monocyte-forming cells in red bone marrow? Macrophages. And a rise in the level of interferon in the body indicates what kind of infection? A viral infection. Okay. This is figure 14-11, which divides immunity up into adaptive or specific immunity and innate or non-specific immunity. But mainly what it's good at, or what it's good for, is showing you the divisions of the adaptive immunity. There's going to be several questions, and I can tell you, I already made the final exam, so I know I asked more than one question where you have to know whether or not, like, it'll give you a scenario and you have to be able to tell, is that an example of naturally acquired active immunity, artificially acquired active immunity, naturally acquired passive immunity, or artificially acquired passive immunity? So let's talk about those real quick. With active immunity, it's going to develop in response to antigen exposure. So you have to be exposed either by being sick, which is naturally acquired active, or by being given a vaccine, which is artificially acquired active. Passive immunity is immunity produced by the transfer of antibodies from another source. So naturally acquired passive would be, for example, the antibodies that cross the placenta from the mother to the fetus, or in breast milk. That's naturally acquired passive. But artificially acquired passive is if you get like an injection of monoclonal antibodies. That was really popular with COVID. So, or the rabies vaccine is really not a vaccine, it's an injection of antibodies. So that would be artificially acquired passive. All right. The adaptive or specific defenses respond to the presence of specific antigens. It's provided by the coordinated activities of T and B cells, or T and B lymphocytes. T cells are active in cell-mediated immunity, and B cells are responsible for antibody-mediated or humoral immunity. The features of adaptive immunity, number one, is that it's not present at birth. This is something that once the child is born, the adaptive immunity begins to develop. It can be active or passive. Active, again, means that it's the response to an antigen. Passive means that your body is not, you're being given the antibodies rather than your body making them on their own. And adaptive immunity can be acquired either naturally or artificially. So artificial would be like a vaccine or an injection of antibodies, an antibody injection. Active immunity is developed after exposure to an antigen. It's a result of a specific antigen. It's a result of the host's immune response to the antigen. Naturally acquired active immunity is due to the exposure of pathogens in an environment. In other words, you probably got sick, and then you developed immunity, so the next time you encounter that pathogen, you won't get sick from it. So that's just from getting sick from that specific pathogen once before. Artificially acquired active immunity is from a vaccine. So a vaccine will stimulate your body to produce antibodies. The principle behind immunization or vaccination is responsible for artificially acquired active immunity. Vaccines are designed to induce an immune response and cause your body to make its own antibodies. And the vaccine contains either the antigen or a piece of it called the antigenic determinant. But it's the piece that your immune cells react to, that they respond to. With passive immunity, this is produced by transferring antibodies or lymphocytes to a person from another source. If it's naturally acquired passive, then it probably is coming to the baby from the mother, either from the mother's placenta or through the breast milk. Artificially induced passive is when antibodies are injected. One of the popular treatments for COVID, particularly in people who were high risk or who had developed a bad case of it, I guess you could say, is the injection of monoclonal antibodies. And the rabies vaccine is actually an antibody injection. It's not a vaccine. It's not a true vaccine. So a true vaccine is exposing your body to the antigen that triggers the immune response. But the rabies shot is actually giving you antibodies. So if you're given a vaccine, it's going to cause your body to make its own antibodies. But the rabies shot, they give you the antibodies. Your body doesn't make them. Now, there are 5 properties of adaptive immunity. One is specificity, which we've already talked about. The T cells and B cells have receptors on their plasma membranes, receptor proteins, that have the ability to match up to or their shape, have a shape similar to the antigen that they're being exposed to. So that foreign antigen must have a receptor on a T or a B cell that can bind to it. And it's specific just to that antigen. So that's specificity. Versatility. There are millions of different lymphocyte populations produced. Each of them have a different antigen receptor. This especially happens when in young children, as the child is growing, their immune system is growing too. Memory is the ability of the adaptive immune response to respond to a second exposure to the antigen, and that second exposure is going to trigger a faster, stronger, longer lasting immune response. So this memory is only a property of the adaptive or specific immunity. And tolerance. Tolerance is very important. This prevents autoimmune disorders. This prevents your body from attacking its own cells. So tolerance exists when the immune system does not respond to normal or self-antigens. It doesn't respond to antigens present on your normal body cells and tissues. Okay, so here we have an overview of adaptive immunity. In adaptive immunity, antigen presentation triggers an immune response by specific defenses. And this is showing you some different antigens. The glycoprotein spikes on a virus, you can see it in both of these top 2 examples. They look like little blue popsicles on sticks. And then you have these little red dots on this bacterium. So those are actually the pieces of the bacterium or the virus. These are the pieces that have receptors on your T cells and B cells. So that's what your immune system is responding to, is just these little pieces called antigens that are attached to the surface of the virus or the bacterium. All right, in cell-mediated immunity, phagocytes are activated, and then those phagocytes, like macrophages, will then activate your T cells. Your helper T cells, specific cells, special T cells called helper T cells, have to also activate other T cells and B cells. And then the B cells are responsible for antibody-mediated immunity. So the response that you get from T cells is to find the pathogen and attack them, either by phagocytosis or the release of chemical toxins and destroy them. And then the antibodies can also destroy antigens in various ways. All right, antigen presentation. This is very, very important in activating the T cells, which then will activate the B cells, other T cells and B cells. So antigen presentation is the process of activating the T lymphocytes or T cells, the helper T cells. Antigen recognition is defined as the binding of the lymphocyte to the specific target antigen bound to receptors on the plasma membrane of other cells. So the antigen on the other cell, the foreign antigen, binds to a receptor on the lymphocyte. That's antigen recognition. When it's able to bind, it's because the shape is similar. It's able to bind because it's compatible. So a T cell that has an antigen recognition site for, let's say, the Streptococcus bacteria that causes strep throat, it's not going to have that same antigen recognition for a cold virus, for example. Then you have antigen binding receptors on cell membranes called MHC proteins, and that stands for, it's a long term, but it's really easy if you think about it, major histocompatibility. So what does histo mean? Tissue. So these are the tissue compatibility proteins, and there's two classes of them. There's class 1 MHC proteins, and all nucleated cells have those. Then there's class 2 MHC proteins that are only found on antigen-presenting cells, which are primarily dendritic cells that are found within the skin and or beneath the skin, and macrophages. Macrophages and dendritic cells tend to primarily be your APCs or antigen-presenting cells. T cells exposed to the APC membrane, the plasma membrane of the dendritic cells and the macrophages. When those dendritic cells and macrophages engulf something and break it down, the antigen portion, they can then kind of push it out to the surface of their plasma membrane, and that's antigen presentation. All right. Then they present that antigen, they come in contact with a T cell, and when that T cell is exposed to that antigen, if it binds to it, then that's antigen recognition. That will activate the T cell and start the immune response. I know it sounds really complicated, but basically just remember this. You have a class 1 MHC proteins on all cells that have a nucleus. So red blood cells that are fully mature would not contain class 1 MHC proteins because they don't have a nucleus. But then class 2 MHC proteins are on only APCs or antigen presenting cells, which are dendritic cells and macrophages primarily. Now, for the activation of the T cell, we also have to include these markers called CD4 or CD8 markers, okay? So T cells have membrane proteins called CD markers. That stands for cluster of differentiation. The type of CD marker determines the response to the MHC proteins, okay? So there's two CD markers. There's CD8 and CD4. Cytotoxic T cells have the CD8 markers, and they respond to antigens on class 1 MHC proteins. I don't know why this helps me, but I remember the number 18, and that means class 1 corresponds with CD8. 18, 1 and 8. You know, I don't know how I remember 18, I just do. And then to remember CD4 in class 2, I remember the number 24. So I remember 18 and 24. And if you can come up with something better, that would be great. I'll share it with the rest of the class, but I remember the number 18 and the number 24. Helper T cells or CD4 cells, they have the CD4 markers on them, and they respond to antigens on class 2 MHC proteins. So you always want to associate CD8 markers with class 1 and CD4 markers with class 2. And we'll go into why and help you understand this a little better as we go through this. But the T cell must bind to a second site for co-stimulation before it's activated. The immune system is designed to be really, really careful before it responds. Because when the immune system does respond, if it's wrong, then you have all these symptoms that you don't need, and it can cause side effects that you don't need. So it's especially important for preventing autoimmune disorders. So the T cell, whether it's a CD8 or a CD4, it must bind to a second site for co-stimulation before it's activated. So first it has a receptor that has to bind to the antigen itself. Then its CD4 has to bind to a class 2 protein, or its CD8 has to bind to a class 1. So those two things have to happen. When activated, T cells will differentiate into several different types: cytotoxic T cells, helper T cells, memory T cells, and suppressor T cells. Just remember, it's the helper T cells that initiate the immune response. Helper T cells have to they have to recognize the antigen before more T cells can be activated and before B cells can be activated.